okay? It was broken up into a lot of different steps on the video lecture. We're just going to do from beginning to end all the different parts. And it follows a sequence. It goes from beginning to end when it happens. So let's label a few things, and then we'll label things as we go along. This is the end of a nerve. All right, this is our axon terminal. Good. So the end of a neuron, end of a nerve cell. And got my orange here. What happens is the signal is sent. We're going to put AP, that's an action potential. This will make more sense when we talk about the nervous system. But essentially, this is the signal, the electrical signal that passes through the neuron and then reaches the axon terminal. Okay, so we got a lot going on here, so we're going to zoom in. Bless you. No. Zombie apocalypse. Starts with sneezes. Okay, so the signal comes down the axon terminal, and what we have here is a voltage regulated, calcium channel. And when the signal reaches the channel, it opens, it swings open here. And because calcium is in higher concentration outside the cell, what's going to happen? It's going to go inside the cell. And what process of transport would that be called? Okay, it would be called facilitated diffusion, because it's facilitated by the channel. Well, sitting inside of this axon terminal, we have synaptic vesicles. And they are filled with acetylcholine. And when this calcium floods into the axon terminal, that is the signal for these synaptic vesicles to empty into our synaptic cleft. Via what process are we? Right, so this is exocytosis, where those vesicles, the membrane surrounding the acetylcholine, fuses with the membrane of the axon, and then it releases acetylcholine into our synaptic cleft. Okay, is this an active or passive transport mechanism? You guys remember? Is exocytosis active or passive? Active, yes. It requires energy for that fusion to take place. So we can put a little ATP over here. All right, on the other side of the synaptic cleft, we have this is the sarcolemma, which is, what's the sarcolemma? Yeah, it's the plasma membrane of the muscle cell. So embedded on the other side of the synaptic cleft are acetylcholine receptors. And so we call acetylcholine, it belongs to a group of chemicals we call neurotransmitters. Because they are transmitting the signal from the neuron to the next cell. Okay, they can also transmit to another neuron. But in this instance, it's transmitting it to our muscle cell. So these acetylcholine receptors will bind to acetylcholine. Acetylcholine will then diffuse across the synaptic cleft and bind to the receptors. So these acetylcholine receptors are also chemical regulated gated channels. What does that mean? That means once the chemical is there, then the gate swings open. And now, what we have is sodium, which is in high concentration outside the cell, goes through the gate into the cell. And this starts another action potential or electrical signal. Okay, again, this is another example of facilitated diffusion. So we have exocytosis, an active transport. We've got two instances of facilitated diffusion. We have one example of regular diffusion. We have a voltage regulated channel, and we have a chemical regulated channel. And we're just getting started. Yes?
Will you scroll up just a little bit? Okay.
Yes.
So the part at the bottom, that's the muscle cell?
Yes.
Okay, what's the orange part?
That's the axon terminal.
Oh, that's
Just not the right color. Sorry. So yes, well that's another good point. So this whole thing is called the neuromuscular junction. And the process that we're talking about right now is excitation. So we're talking about excitation at the neuromuscular junction, which starts with the action potential coming down the axon terminal. So let's just number through these. Give myself a unique color here, purple. Here's step 1. The action potential. Here's step 2. The channel opens, calcium rushes in. Here's step 3. Calcium starts to signal for exocytosis. Here's step 4. Neurotransmitters bind, sodium rushes into the cell. All right, this action potential is then propagated along the sarcolemma. And then goes down our T-tubule. All right, on the T-tubule then, we have I'm just going to color this in. Voltage regulated, we've already had this, so I'm abbreviating now. Gated channels. Which open when this signal passes from the T-tubule into the, what's this dumbbell thing? Sarcoplasmic reticulum. Right? And what is this structure then that we learned last time? The triad, right? Which is made of one T-tubule and two terminal cisterns. So what happens when these channels open is calcium from the terminal cisterns and from the T-tubule go into the cell. Yeah, so the signal, which started up here, this action potential, which then traveled down the T-tubule, then causes these voltage-regulated gated channels to open, and calcium from the triad enters into the muscle cell. Yeah.
You drew calcium going from the T-tubule directly to the sarcomere. It doesn't have to go through the sarcoplasmic reticulum first.
No.
Okay, so they have their own channels that go into the sarcomere.
Yeah, some of the calcium comes from the T-tubule, some from the terminal cistern. Both are stimulated by this
So then
voltage.
The only purpose for the channels going from the T-tubule to the cistern is to propagate the action potential?
Yeah. There's channels on both. The signal is passed from the so I kind of drew two things at once.
Right. So, but
Signals passed from one to the other. Both have their gated channels that open.
But the channels between the sarcoplasmic reticulum and the T-tubule are not to transfer calcium. They're to
Right.
propagate
There's
the
no
action.
calcium being transferred, yeah. Okay, now underneath this, although I haven't drawn it underneath like we did last time, we have some recognizable players here, right? What is this? This is our thick filament, myosin, and our thin filament, actin. And in black here I drew troponin. And in red here, tropomyosin. So at rest, the binding sites are being blocked by the tropomyosin. So there's no interaction between actin and myosin. But when calcium comes and then binds to tropomyosin, it shifts. And then the binding sites are available, and the actin and myosin will start interacting, which we'll show in a second.
So the calcium helps it shift?
Calcium makes it shift, yes.
Like takes the tropomyosin off of the actin.
Yeah, it reveals or exposes the binding sites for the myosin to bind into it.
So then the tropomyosin form, what?
Okay,
I mean, like the tropomyosin forms with the actin. Is that what you're saying?
so yes, the tropomyosin is on the actin. So we've got these binding sites here, right? That these myosin heads could fit into, but they're blocked by this tropomyosin. So when this calcium signal is released, when this calcium is released into the cell, it binds to the tropomyosin and then it shifts it so that those are then revealed. So here, I'll erase some of it. So if calcium binds, then it's going to move up here. And these binding sites are now exposed and available. And now myosin can fit right into there. Bind, and then contraction will initiate. All right, so let's do some more numbering. So we have our action potential going down the T-tubule, release of calcium, binds to the tropomyosin, and then we have contraction. Up here. You could break it up into more steps than that. We're just combining a few steps at each location. All right, so this is when the signal from your brain is causing contraction in the muscle, okay? So if you're flexing your muscle, that's what's going on. But when you stop flexing, or when the signal stops, okay, then we have relaxation. All right, let me see if I can. Let's do this maroon. That's not how you spell relaxation. But just relax, okay? All right, so relaxation is when the signal stops. So if this signal stops, that channel's going to close, okay? But one of the problems we have is there's still a bunch of neurotransmitter in the neuromuscular junction. So we have an enzyme called acetylcholine esterase. And it's a mouthful. It clears out the acetylcholine. Okay, so it takes it, unbinds it from the acetylcholine esterase. Unbinds it from the acetylcholine receptors. So then, if that happens, then these gates are going to close again. So then this signal will stop. So then these gates will close. But we still have all this calcium here. Okay? And so calcium is actively transported back into the sarcoplasmic reticulum. Okay, so that's going to require ATP. And once enough of it is actively transported, then this shift will go back down. And it will block those binding sites, uncouple the myosin from the actin, and the muscle will relax back to its original shape. You say it makes sense? Yep. Okay. So all of this happens every time you move everything.
And how long does it take for it all to go?
Just instantly. Yeah, that long. I mean, it seems like that would take longer, but the cellular processes happen pretty quickly.
So like cramp, what is that?
When you cramp,
Why can't you release then?
I'm not. So we have a lot of ions involved in this process, right? Cramping, they think, is associated with some sort of ionic imbalance. They aren't sure exactly what causes cramping, but some sort of ionic imbalance where If you can't get the calcium back up, maybe, then you're stuck contracted. So that's why, football players who are playing, in late August or early September, they're drinking something that has lots of ions in it, like Gatorade or pickle juice, apparently, prevents cramps. I don't know the science behind that or if there is science behind it, but apparently it works. But it would be something high in electrolytes. All right, so that's excitation to contraction. That's easy, that's nothing. But it does follow a step, right? It does one thing after another. So if you go through it enough, you should be able to tell the story. Okay? But there is one part we didn't really talk about, and that's what's going on with the actin and the myosin. Okay, so we'll do that right now. So once the signal is all there for contraction to continue, this is what is happening during contraction. So the first thing that happens is a cross bridge. Is formed, okay? And that's just where contact is made between the myosin head and the actin binding site. All right, and what happens on these myosin heads is they have enzymes for breaking down ATP and using ATP for mechanical movement, right? So at this point, it's already broken ATP. Zoom in. It's already broken ATP into ADP and P, right? So ATP has 3 phosphates on it, right? Okay, and then when you break off one of those bonds, you release energy. And so at this point, we have now ADP, which has 2 phosphates, and then the other phosphate, which is broken off. Okay, so we use some of that energy to make this cross bridge. But we've just made contact at this point. Our next step, we have moved it along, right? So we went from this position to that position. This is called our power stroke. Okay, this is where it actually moved, right? And when this happens, we've used up all the energy from ATP, and our ADP and P are released. All right, but now we're stuck. We're stuck in that position until we get another ATP. Okay, so this third step is detachment. And we need an ATP in order to do that. Yeah, ATP. And then to get into our high energy configuration, which is the cocked position. So pulled back, ready to go. We're going to hydrolyze that ATP, use some of that energy be ready to go. And because there's tons of these, right? Myosin heads on here. And so when this one attaches, ratchets, the other one's probably detaching and getting into the cock position, they're going to all take turns and essentially climb up the actin, and that's going to cause those A bands to come closer together, the H zone to shrink, and the distance between all those sarcomeres to condense, and that's what makes your muscles move. All right, you guys got it? All right, so go ahead and do 5 push-ups, just get it in your head, like that's what you're doing. And then we're going to do it again. Right, well actually, yeah, it'll get stuck on the power stroke and can't go to detachment. You need ATP in order to detach. The ADP kicks out after it moves, after it does the power stroke.
But in order to detach, it has to have an ATP.
Right. So it can go and ratchet for a while. It can go through a few of these steps. Once it runs out of ATP, then it's stuck and it's attached. And the calcium can't be pumped back. Right? Okay. All right. Let's see. We have 2 conditions, which we talked about in the video lecture. Myasthenia gravis and rigor mortis. All right, so where in this, we don't have all the details on this, but where in this steps of excitation would myasthenia gravis have an effect? Do you remember what myasthenia gravis is? Okay. This is an autoimmune disorder. Yeah. So, what is an autoimmune disorder? Yeah. This is when your white blood cells, your immune system, attacks some cell or tissue or something in your body. Okay? What cell, tissue, protein do you think myasthenia gravis has an effect on? What's that? Myosin, good guess, but that's not it. All right. It is actually our acetylcholine receptors, which we drew like this funky shape. So if your white blood cells are in here eating up these receptors, what would happen? Yeah, the signal wouldn't pass very well into the muscle, okay? And so depending on how many of those are damaged, you're going to have problems contracting muscles. So yeah, and an inability or an ineffective contraction. So there's a lot of neurological problems which happen in advanced stages of myasthenia gravis, including confusion, weakness, fatigue. Drooping eyelids is one of them, where they can't keep their eyelids open because the signal to do that isn't there. All right, so that is where myasthenia gravis affects this process. All right, what's rigor mortis? All right, so this happens after something dies and the muscles get stiff. I don't know if you've ever felt something after it's died, like a bird or a squirrel or something. What? Well, your minks, They are pretty stiff, but they aren't in rigor mortis. It happens kind of immediately after your death, but then wears off hours after. All right, so what happens there is when you die, or when something dies, the infusion of ATP decreases. Okay, so if, and this calcium, right? This calcium is being held there by ATP. And also, these myosin heads need ATP. So after you die, this cellular process starts to break down, and calcium just flushes into this system without the signal. Okay, that causes then the actin and myosin to interact and to ratchet. But if there's no ATP, what step is it going to get stuck on?
Because does ATP go in and kick out the ADP?
And so the muscles are stuck in a contract position, and that's why it's stiff.
Isn't
Yep.
that only for like a short while because ATP does degrade?
So yeah, after a while, the connection between the two degrades, and then it softens up. Yeah. So rigor mortis is not a permanent condition, but shortly after death, it should kick in, and then hours later, it will start to relax. Okay, so you should know then where these things that we talked about affects different parts of the excitation process. All right, so can you guys do it again?